This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, a special episode recorded on October 29th, 2014. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. I have a special guest for you today. He's professor of genetics and molecular microbiology at Tufts University, also director of the HIV drug resistance program of the National Cancer Institute. John Coffin, welcome to TWiV.
Thank you very much. I'm delighted to be here.
It's great to have you. I know over the years of the XMRV story, we corresponded a bit. I saw you at a meeting in Boston, so it's good to have you here. You're visiting today to give a seminar, and I always like to grab people when they're here. I'd like to talk all about your career and your science as much as we can cram in an hour and a half. I want to start all the way at the beginning.
Okay.
Where you're from, New England?
I was born in Boston. My father was a fellow at MIT in materials science at the time, and then was recruited by General Electric, which we actually called in the family Generous Electric, to go to their headquarters in Schenectady, where they have a big research lab in Schenectady, New York, upstate. And so I was raised there. My family lived there for quite some time, actually.
And you went to high school there?
I went to high school in Albany, which is nearby. And then I was an undergraduate at Wesleyan University in Middletown, Connecticut. And from there I went to graduate school, and I was fortunate. This wasn't my choice. It was sort of an accident, but I ended up in the wonderful laboratory of the late Howard Temin.
Had you always been interested in science?
I had always been interested in science. I had always planned to be a scientist.
Do you know why?
No, I was just interested. My father, of course, was very technically oriented. He was an engineer. He studied things. He did various different stuff.
Yeah.
Although we ended up interacting in a way that I'll tell you just a second, which you'll find interesting. He studies material. He studied materials and why metals break, why jet engines fall apart, or why nuclear plants develop cracks that cause serious
maybe
leaks
ultracentrifuges too. Right?
And it could have been ultracentrifuges. I don't think he worked specifically on ultracentrifuge, but it was exactly the same kind of issue. Yeah. And he and I, so our careers, of course, were scientifically couldn't have been more different. Except there was one place where we actually came very close to one another, and that was on the pages of Current Contents.
Of course.
back when there was a Current Contents.
I remember that. They actually made a paper version.
And that was those were bound into big books every year. Yeah. And so he recognized that he would go check himself out every now and again in this, and see how many people were. Actually, it was a Science Citation Index. I'm sorry, more than the Science Citation Index. He'd go see how many people had cited his work. And then he noticed that there was this other Coffin kind of creeping up on him. Well, his name was Lewis, and it starts with L, mine's J. So we're actually quite close together in these pages. And I eventually caught up to him and overtook him by virtue of a review that I published that got a lot of attention. And
that's funny.
And turned out to be fairly well enough cited so I could pass my own father in the pages of this.
What's the ancestry of your family? Do you know?
My distant ancestors were in Cornwall in England. The first Coffin that came to the U.S. actually was a fellow named Tristram. He came to Newburyport, which is a town north of Boston, and he owned a tavern. He was from a fairly wealthy family, but he was a second or third son or something like that, so he wasn't due any inheritance, so they sort of shipped him off to the colonies. This was the mid-17th century, I think. And then he and some friends, some buddies, called Macy and Starbuck and Folger, names you'd recognize if for nothing else because of the coffee connection. They got together and bought the island of Nantucket from the British Crown. And in fact, the Coffin family became quite prominent on Nantucket.
Wow.
A bunch of whalers And he had 8 sons, all of whom have surviving descendants today, so the family spread out quite fast.
And you're from that family?
I'm from that family.
Nice.
So it's easy to... I very frequently run into very, very, very distant relatives. We're still talking the 17th, 18th century.
Yeah.
So it was a long time ago.
So why did you go to Wisconsin? Did you know the science there?
In part, I knew the general reputation. I applied to a bunch of graduate schools, and of the ones I was accepted at, this one looked the best to me. I was somewhat I had some vague interest in cancer research, although not very well formulated. And the McCardle Laboratory at the University of Wisconsin was a very strong center for cancer research at the time, and so that was a real attraction. And that was the graduate department I applied to.
When you went to Temin's lab, was he in the throes of discovering RT, or had it already been done?
No, it was before that. He was
that.
in the throes of actually receiving a lot of grief for constantly arguing that the retroviruses must replicate through a DNA intermediate. And he was widely disbelieved. Even his former, actually there was quite a potent feud between him and his former fellow postdoc in Harry Rubin, with whom he published some of the first papers on developing the very important quantitative assays for Rous sarcoma virus based on the ability of the virus to transform cells in culture. And he and Rubin ended up feuding about whether the provirus existed or not. And Howard published quite a number of papers claiming evidence for the existence of the provirus. But when you look at them, you could easily not believe them. I mean, the evidence wasn't all that strong that he had. There were things like hybridization experiments where he would take labeled virus RNA and hybridize that to DNA from infected and uninfected cells and get a couple of counts more in the infected cell DNA than the uninfected cell DNA. And there were sort of major problems with this besides just the low counts. The fact was you couldn't get very high specific activity nucleotides at the time, so you couldn't label things very well. And one thing he didn't realize and wasn't discovered for a few years after that was that chickens generally contain endogenous viruses, DNAs that are very closely related, of course, to that of RSV, and that therefore that gave a very high background in these assays. So you'd only get a very small differential, and one that people could easily blow off as being not meaningful. And then he did a number of other largely genetic experiments. I mean, the provirus hypothesis came about from a piece really of genetic intuition on his part, where he recognized that if you got different strains of virus, those could give rise to transformed cells that had somewhat different phenotypes. Cells basically had different shapes. Sometimes they were long, and sometimes they were elongated, sometimes they were round. And this was a heritable property of the virus that caused a heritable change in the cells. And he intuited from that the only way this could be happening is if the virus was able to cause a permanent genetic change in the cell. And the only way it could do that, the only reasonable way it could do that was by inserting its own genetic information into that of the cell. And because at the time it was already known that these viruses had RNA genomes, that implied that there must be some way of transferring the information, genetic information from RNA to DNA. Not everybody shared his vision, if you like, on this, and he would often come back from meetings at places like Cold Spring Harbor, for example, quite depressed from the reception that these ideas got. I suppose it's a strategy that if you really believe in yourself, maybe he believed in himself strongly enough that he actually was applying this, sort of using weak experimental evidence to support a brilliant idea, and then eventually coming through with an experiment so dramatic that everybody just fell over, just blew everybody in a way, and then you're locked for a Nobel Prize, which of course is exactly what happened a few years later after he and simultaneously David Baltimore took this and were able to identify the enzyme reverse transcriptase in the virions
So
of retroviruses.
Where did he get this idea to look for the enzyme in the particle?
It probably came from the work of the Baltimore lab. Of course, it was Alice Wong and David Baltimore's lab, and who first discovered RNA-dependent RNA polymerase in vesicular stomatitis virions.
Okay.
And that probably built on the work of Cates and McCausland, who discovered RNA polymerase, DNA-dependent RNA polymerase in pox viruses.
Right.
And so that sort of generated the gestalt that there would be nucleic acid synthetic enzymes in virions, which probably nobody really thought much of before that. And then it became, David Baltimore eventually very nicely codified that, of course, in the concept of positive strand viruses, negative strand viruses, and so on and so forth. And so, but it was quite a dramatic time, actually. This was 1970. It was the height of the Vietnam War, protests. In fact, during that summer, there was a bombing at what was called the Army Math Research Center in a building in Wisconsin, because it had the name Army on it, I think. And a postdoc was in fact killed in this bombing. It was right after the discovery of reverse transcriptase, but it was at a time when there were huge student protests. It was around the time of the shooting of the students at Kent State. It was at a time when University Avenue, which runs outside of where the old McCardle lab was in Madison, was filled with student rioters, or well, not quite rioters, but protesters marching up and down, and then with the smell of tear gas always rolling around as police trying to control this. So it was quite an interesting time, shall we say.
What did you work on in this lab?
So I worked on Rust sarcoma virus, on more peripheral. Nothing as central, unfortunately, as reverse transcriptase. Although I did play a very important role in the discovery of reverse transcriptase, I have to tell you. And that is that the enzyme was discovered by Satoshi Mizutani, a postdoc in the lab, who's a very good nucleic acid biochemist. And he or Howard, I'm not sure which one, realized that in order to be able to assay this enzyme in virions, you had to be able to break the virus open in some way that didn't destroy the enzyme. And so he came to the idea of using some non-ionic detergent or another. Well, I happen to have a bottle of Nonadet P40, and the only one around. It was actually a little hard to get at the time. You had to write specifically to Shell Chemical Company, who made it, and they would send you a bottle for free, but you had to ask nicely for it. And so I had the bottle, I had asked nicely for it, and that's the bottle of detergent that Satoshi used to discover reverse transcriptase with. So I've always been quite proud of that.
It's been renamed now, I understand.
NP40?
Yeah, there's something else.
Oh, really?
I don't know what. Yeah. Huh.
Yeah, that I didn't know.
So you didn't work on the RT?
So I didn't work. I worked on RT afterwards. I did some experiments which actually turned out to be sort of misguided based on what we learned later about how retroviruses are made, and to look for RT containing particles that contained reverse transcriptase in cells that were making virus, and with the idea of trying to then see if we could find that, if we could develop a good assay, then maybe we could also go looking in cells that didn't have virus in them to see if there was any role for reverse transcription in normal cellular processes, which was an idea that Howard had at the time, which was perfectly rational, but turned out not, well, with the exception of telomerase, which is a different kind of RT, which we wouldn't have been able to find with that kind of assay. But and then I also worked a little on the phenomenon that mammalian cells infected with this virus become transformed. They express a Sark gene, which we didn't know about at the time. But they don't. But they don't make virus.
Right.
And so I did some experiments showing how the process by which you could rescue virus from these cells, if you fuse them with permissive cells, how that happened. And that was basically my thesis.
What was it like to work for Temin? I never met him, but I hear
he was a very interesting guy. He was very mild. He came across as being very mild-mannered, but he could also be very cutting. He could make very incisive comments. And he would say what he think. He would not be afraid, no matter in almost any circumstance, to say what he thought. I mean, there's a famous story, of course, that you probably know and you may have recited a few times. When at the Nobel Prize dinner, or the dinner after the Nobel ceremonies, big formal dinner and so on, when I think everybody was supposed to give a little speech, not the real Nobel lecture, but a little speech. The first thing he did was sort of thank all the assembled nobility present by asking them to please stop smoking. Put out all their cigars, and he used the occasion to give a harangue against smoking.
Oh my gosh! Yeah. I didn't. I might have heard that. I forgot.
He always said you have to seize the opportunity. If there's something you really believe and you want to do it, you got to seize the opportunity when it shows up, and he certainly did it in that case.
So as a mentor, he was attentive.
Oh yeah, very much so. He was. We had you. He did something I've never been able to do, and I think a few faculty I know have been, and that was he would have very regular meetings with everybody in the lab every Friday afternoon. We used to call it Friday afternoon confessions, and he would have sitting on his desk. He'd have a series of slips of paper, and every time he thought of something for any one student, he'd write it down on this little slip of paper. So you'd go in, and there'd be this little stack of paper with your name on it, basically, and he'd go through through all the things that he happened to think of during the week. So I've never been able to be that organized with my students.
Right. I heard he used to sit in the front row at the Cold Spring Harbor meetings.
He always sat in the front row. That I do. So
you still do that?
He always sat in the front row of the Cold Spring Harbor meetings. He would almost always go up to the speaker after the session and offer words of advice and encouragement or whatever. He was very, very good about that. He was always And
he asked a question of every speaker, right?
Yeah.
Pretty much.
He always had a question.
It's interesting. So Steve Goff says you have some great stories. Can you remember any of Howard Temin? I don't want to put you on the spot, but just in case you remember any of them.
Well, he was never socially really smooth. He always came across, although he was much better than he seemed in some ways, but he always came across as being a little awkward socially. But he was always so sincere. And I'm trying to think of a specific. There was okay. He did teach me something important. At one point in the course of some of the experimentation, we were trying to get some anti-sera against the virus we were working with, and neutralizing anti-sera. Well, the only way to make neutralizing anti-serum against this virus was to get it from the natural host. You couldn't if you inject the virus if you just took a preparation of virus and injected it into mice or rabbits or something, you'd get a lot of antibodies, but none of them would be neutralizing. So you had to do it in chickens, which was the natural host of this virus. And so we had a couple of chickens in the lab. And so I injected them and waited a while. And then time came, and he was going to show me how to properly bleed a chicken. He bleed a chicken by cardiac puncture, actually, which is a skill that I never really mastered completely, although I did manage to had a few successes. Well, while we were doing this, one of the chickens got loose, went flapping down the hall into the next lab. Well, the next lab was that of Vaslav Shabalsky, I'm sure you know.
Yeah.
And in particular, it ended up on the desk of Fred Blattner, who you probably who's a postdoc in that lab at the time, who you probably also know, and many of your listeners probably know.
Yeah.
And it was really quite. And Howard was the one that showed me how you properly catch a chicken.
That's great.
So
chickens running around.
That was one of the especially memorable moments, I would say, of my graduate.
I guess you caught it, right?
We caught it. Yes, we got it back again.
So after Temin's
Fred
lab,
was not amused, actually.
Yeah, I'm not surprised. Yeah. After Temin, you went to Charles Weissman for a postdoc, and what did you work on there?
I started working on with a couple other postdocs there on the mechanism of retrovirus, again sarcomavirus, the mechanism of RNA synthesis. This was just after reverse transcriptase had been discovered. So it had been discovered that the virus is capable of encoding DNA. It kind of followed from that further replication, and the provirus model had that the DNA would then be integrated into the host cell genome, although nobody had been able to demonstrate it to that point. And that further replication should be carried out by host cell RNA polymerase. And that, in fact, was what we first worked on. We were able to show that that was true. And furthermore, that the enzyme was RNA polymerase 2 based on its sensitivity to alpha-mannitine, which is a selective inhibitor. And we were able to develop a system where we could get synthesis of virus RNA in isolated nuclei from infected cells. And one of the reasons we were able to do all of this is that at the time, the Weissman lab was worked mainly, although they had a retrovirus project going for some time, their main effort was in bacteriophage Q beta, and in vitro replication of that bacteriophage, which actually has some very interesting aspects. And Charles had come from Severo Ochoa's lab here in New York at NYU, and who was a very good nucleic acid biochemist. So that was an advantage. That's why one of the reasons I went to that lab is I really wanted to get some really good nucleic acid biochemistry, which they could do. One of the things that they did was they made all their own labeled P32 labeled nucleotides. Fortunately, the retrovirus people were considered, I think, too unskilled biochemically to be allowed to use the 500 millicuries or so of P32 that were necessary to make one of these preparations of labeled nucleotide. But the availability of those actually was a huge plus in being able to do some of these early experiments on RNA synthesis, because they were not very widely available at very high specific activity commercially in the early in the early 70s. And so the first things we did were demonstrating that the virus RNA was made in the nucleus. It was made using RNA polymerase 2 with the normal host systems, all completely consistent with the provirus hypothesis that Howard had put forward initially, probably 10 years before that. And then moved on. Since the lab was very good at labeling RNA, we could make large amounts of P32 labeled RNA to develop and apply a fingerprinting approach, which digesting the total RNA with T1 ribonuclease, which leaves small fragments, all of which terminate in G, because T1 cleaves only next to G. By chance, a few of those will be quite long and to be unique. To be unique in composition, and you could separate them with the 2D gel system, and and so we use that fingerprinting approach to answer a question actually which had come up when it was discovered that retroviruses had the RNA genomes of retroviruses were present in multiple copies, and that's yes.
So I'm showing you my thesis.
Yeah, there you go. That's exactly right.
That's what I did too.
Yeah, that's a 2D gel, isn't it?
It's a 2D gel. It's influenza virus RNAs. And we had to make our own gamma P32 ATP and used it to label the T1 products.
Oh, you did it that way. Okay, so we labeled with P32.
In infected cells, right?
And in infected cells, and then which was hellacious from the amount of P32 you ended up throwing around.
Yeah, that's why we didn't do that.
Although I spent a lot of my early years as an assistant professor also doing the same thing and using viruses that grew even worse, requiring still larger amounts of P32 to label large enough amounts of P32 that you'd buy it and the bottle would be already brown from the effect of the radiation.
We used to have a standing order of 50 millicuries a week.
Yeah, we would get, sometimes we'd get as many as 500 millicuries.
Wow.
I had a postdoc who was working with a really badly replicating virus that would give. And he would sometimes label with half a curie.
Wow.
Of P32. It's funny now, actually. Students, a few microcuries, and they'll be
nuts.
carrying it around in this big shield. And I think people are really
I feel the same way. Yeah.
People are really quite silly about that, in my opinion.
I know. When I came here, a few microcuries would freak everyone out. I said, "Are you serious?" Did you go right to Tufts after?
I went right to Tufts from there.
Oh, so you've been there your whole carear.
Yeah. That was 1975.
75.
So that's going on. It's going on 40 years.
I had just gone to. I was just in Peter Palazzi's lab in 75, right? And I went to Boston in 79, but I never made it over to Tufts. Who hired you? Did Elio Schechter hire you?
Elio Schechter, yes.
He's on one of my other podcasts.
Yeah, right.
I mentioned you once. He said I hired him.
Yeah, he did. He did indeed. He and Satver Tavethia.
Yep. The Tavethias, I remember.
So anyway, we actually were the first ones to show how big the virus genome was, because it wasn't known to within a factor of 2 or 3, because it was known there are multiple copies of RNA.
Yeah, right.
But it wasn't as there are in influenza.
Yeah.
In influenza, of course, there are 8, and it's pretty obvious each one of them is different. And in retroviruses, it wasn't at all clear whether the RNAs, it turned out to be 2. Actually, it was thought to be 3 at the time, because counting wasn't very good.
Yeah.
That whether they were the same or different in sequence, and we were able to show that they were the same in sequence by showing what the overall complexity of the RNA was, how many units there would be in a single genome's worth of RNA.
Right.
And the answer was quite clear that there was that the complexity was about 3, equivalent to about more or less 10,000 nucleotides.
Mm-hmm.
And I remember when I first told Howard that, he was very surprised that the virus was that small, actually.
It's amazing what you can do with small genomes.
It's amazing what you can do with small genomes.
So when you started your lab, what problems did you work on?
One of the things we first looked at was the mechanism of reverse transcription. I actually collaborated a bit with Bill Hazeltine, who you probably also ran into when you time in Boston, because you were in the same lab.
I heard stories about Hazeltine, because he was a former Baltimore postdoc.
He was.
He had left behind many stories.
Yes.
Like running ultracentrifuges for days filled with water so he could have them ready.
Just so they'd be ready to go in case he needed them. I've heard that story. And I hope he's not listening, but he probably not. Probably isn't.
Probably not. He's far removed from virology.
And but one of the first things we did, it had been discovered by somebody else, I think principally John Taylor. It had first been discovered that the virus RNA synthesis starts with a tRNA primer, which has been a little bit of a surprise. Some very nice work. And John Taylor, I believe it was, had discovered that the tRNA primer was located in a surprising place. You would have thought it would have been somewhere near the 3' end of the genome, well posed so that the whole genome, you could start DNA synthesis at that point and copy the whole genome. Well, no, that wasn't it at all. It was way down near the 5' end.
Right. Yep.
And Bill had been working on looking at the products of reverse transcription in a test tube, because you can make reverse transcriptase go just by taking virions, adding some detergent, adding nucleotides. The enzyme that's already in the virions will start to copy the RNA that's already in the virions, and you can look and see what the products look like. And what he found was you could get a series of products of varying length, but seemed to be a fairly well-defined size, which were interpreted as the enzyme moving and then stopping, waiting a while, moving on, stopping and waiting a while. Well, there's one product that's particularly prominent called strong stop DNA that was about in this virus about 100 bases, different lengths in different viruses, and that you could show fairly easily when it was made was actually connected to the tRNA. So this is the first piece of DNA that's made that's connected to the tRNA. Right. And so what we were able to show was two things. One is that the strong stop DNA recognizes if we took the strong stop DNA and hybridized virus RNA to it, we could find protection of a sequence, hybridization of a sequence that included the capping group, meaning it was hybridizing to the very 5' end of the RNA, meaning that what this sequence represented was starting at this tRNA molecule, which is our primer, which is already very near the 5' end, and just running to the end and stopping.
Right.
And but then we went on to show that this product also hybridized to the 3' end.
Right.
And that meant that the virus genome was terminally redundant, that it had a little sequence that was present next to the poly A at the 3' end and at the 5' end, and gave rise to what was then and has often subsequently been called the jumping model of reverse transcription, where it copies from the end, from the tRNA to the end, stops. It had been discovered originally, I think, by Karen Merling, that the enzyme also has associated with it a ribonuclease H activity, which degrades specifically RNA when it's in an RNA-DNA hybrid. That cleaned off the genome that had already been copied, and that allowed the end to associate with the other end, and that allowed the DNA synthesis to continue.
And that process we know now is needed to generate the 2 LTRs.
That process that needs to be done again, basically going the other way, and that was done originally by in David Ballimore's lab by. It'll come to me. I know Steve Goff was involved, but it wasn't Steve, I think, who did it. It was somebody else.
Is it Ellie Gilboa?
Ellie Gilboa, thank you very much.
Yeah, so that was going on when I went. Steve and Ellie were there.
So that was starting with that jump. Ellie showed that another jump had to take place. And it's a combination of those 2 jumps that gives rise to the LTRs at either end of the viral DNA.
Did you get to know Steve at that point when he was?
I knew Steve a bit at that point. Yes.
Okay. And then probably more when he came here and had his own lab and so forth.
I've had many contacts with Steve over the years. We see each other all the time, here and there.
Yep. Well, you could be his brother. You look very similarly.
Well, yeah, except he's bleached his hair.
So one of the things you've worked on for many years in your lab, which I wanted to talk a little bit about, is endogenous proviruses in mice in particular. But I wanted to just, how did you? Well,
we actually started out working on endogenous virus of chickens.
Chickens.
Those were the first projects. I had set up a collaboration with Harriet Robinson, who was at the time at the Worcester Foundation.
Right.
And she was an expert in using chicken models for avian leukosis, it was called leukemia, of birds, and. So it was known already that one of the endogenous viruses of chickens, a virus called RAV0, RAV is an acronym for Rous-associated virus, was originally discovered as viruses associated with Rous sarcoma virus. RAV0 was a virus that you could isolate from certain strains of chickens, and that you could grow that fairly well, although it didn't grow quite as well as the exogenous viruses, but at least in her models didn't cause you could infect chickens with it.
So let me just clarify something. So you mentioned exogenous,
right?
So these are retroviruses that infect, but they're not endogenous in the genome of the organism.
They're ones that are transferred from one animal to another.
So Rous sarcoma is an example of that?
Rous sarcoma would be an exogenous virus
because it does not exist as a provirus in the chicken genome, right?
Right, exactly.
But the
related viruses do.
The leukosis viruses, from which Rous was derived, right?
Leukosis viruses are transmitted exogenously and are found as endogenous viruses. Okay. You get some of each. They're distinguished in their receptor usage. The endogenous viruses in chickens tend to all utilize the same receptor, which is usually not found on most chicken cells. There are some that have it. You can find some lines of chickens that have it, but many don't. It's what's called a xenotropic virus, which is a very interesting phenomenon we've worked on a little bit, where an exogenous virus can no longer infect the species that it finds itself in. You see the same phenomenon in mice and cats and other species. And what has probably happened evolutionarily is that this virus came into the species somehow, we don't know how, and was infecting was infecting this and being transmitted as an exogenous virus, one animal to another, and was putting some selective pressure on the birds.
Right.
At the same time, some of these entered the genome and became endogenous by infecting germline cells. But at around the same time, probably, there was selection for resistance, and the resistance was for mutations in the receptor for this virus that the virus could no longer recognize.
So probably there was an effect of the infection on the animal. Which
there probably was. It was probably causing leukosis or some other disease. These viruses cause a variety of diseases in addition to leukemias. They'll cause a wasting disease. You can get variants that cause osteopetrosis, big ugly looking bones in the bird, and so on, and a few other things. And so they're not, these viruses are generally not really benign. But interestingly, the Rav0, the virus that, and this was one of the sort of first kinds of projects I set up when I set up my own lab. What we showed was that the major difference, besides the receptor it used between Rav0 and these other viruses, was in the LTR. In particular, we were able to show that genetically that it was these differences in the LTR that related to the efficiency with which this virus replicated in culture, and also its ability to cause disease. So that led to the importance of LTR sequences in causing cancer in particular.
Right.
Led to the idea that that cancer might arise by integration of the virus, by integration of a virus containing a strong promoter of some kind, a strong activator of transcription, in the vicinity of a cellular oncogene, or proto-oncogene it was called, a sequence that was capable of being abused in some way to cause that cell to turn into a cancer cell. So we hypothesized this. We unfortunately were trying to do the experiments, but somebody else got to them first. A common theme in science, I'm afraid to say. Sorry to say. Was able to show, in fact, that was the case. It was shown originally, particularly by Bill Hayward in Saburo Hanafusa's lab here in New York, and also at around the same time by the Bishop Varmus Laboratory in San Francisco, that if you looked in avian leukosis in infected birds at the disease they get, which is a B-cell lymphoma, that you could find characteristic proviruses, new proviruses, clonally, that is to say every cell in the tumor had them, integrated next to a specific gene. In this particular case was a gene called c-Myc, which had previously been identified as being in an oncogene-containing virus called myelocytomatosis virus. And so this was really the first direct demonstration that a cellular gene could be directly activated in some genetic rearrangement way, in this case an integration of a provirus next to it to cause cancer. As I say, we were trying to do those kinds of experiments, but we did not, we were probably a very, very distant third in this.
So so if I go to the store and buy a chicken, that chicken has endogenous avian retrovirus.
It certainly does, yes.
Does it have any retroviral particles in it?
Some might.
Some might, depending on the strain.
Depends on the strain of chickens. Some lines of chickens will spontaneously express infectious virus. Others, interestingly, will express just a few virus proteins, and in particular, you'll often get birds that express just the virus envelope protein. And in fact, what that does, and you see the same thing in mice and also in cats and probably some other species, and what that does is it blocks the receptor and actually protects that animal against infection by a virus that uses the same receptor from outside.
So that's been maintained evolutionarily for that purpose.
It's been maintained almost certainly for that purpose. And if you look at some of those viruses, the other genes are pretty much messed up, but the envelope gene is still good.
But these chickens don't have any disease as they grow up on the farm, right? They're fine. They don't have leukosis or any sarcomas or anything like that.
Well, if they did, the bits that had it would have been chopped off the chicken before it got to you.
They're probably slaughtered quite young before they would.
Most are, but actually, oncogene discovery. A fellow named Peter Vogt is a good friend out. You probably know.
I know Peter. Yeah.
Out on the West Coast. made a career, well, part of a career, out of discovering new oncogenes by going to slaughterhouses, particularly slaughterhouses where they, for some market or another, would slaughter relatively old chickens, and looking on the floor for parts that had been cut off the birds as they went down the assembly line, discarded on the floor.
Yeah.
Many of those parts had tumors on them, and they would take those tumors back. In many cases, extract the virus out of it and give it a good home in the lab. And quite a number of interesting viruses were discovered this way.
Different avian retroviruses.
Right, different avian, particularly transforming ones with new oncogenes.
Interesting. So these probably infected birds millions of years ago, I would assume.
Probably not too many millions of years ago. This particular group of viruses is probably fairly recent. There are much older, there are ones that are fairly closely related that are much older than that.
Right.
But they have, most of those are no longer active in the sense that all of the genes in them are mutated. Although one of them sort of reappeared partially as a recombinant with one of the more recent viruses that gave it a new envelope gene, and this actually was quite a devastating virus in certain breeder chicken flocks for a while.
So recombinant of two different
between an envelope gene that came from one of the much older viruses.
I see.
And the rest of the genes, GAG and POL, the LTR, and so on, from a much more recent virus that occurred in some chicken somewhere.
Right.
And there are certain things about, these viruses are largely transmitted from mother to offspring. They're expressed at very high levels in the oviducts, for example, so they infect the eggs as it goes by. But they can also be transmitted in bloodborne transmission, and there's a lot of the use of the same needles to inoculate lots and lots of birds with the same syringe, basically, for vaccination purposes.
Yeah, sure.
In inoculating vitamins and things like that.
Well, vaccines too.
So the virus, yes, exactly. So
Marek's virus, influenza,
So the same kinds of things, and so these viruses have been spread around that way.
That's interesting.
As have many other viruses.
Do you know this story of the reason why some chickens have blue eggs is because a retrovirus integrated next to the gene for the pigment? Right.
So retroviruses, endogenous retroviruses can in fact do that. The reason that starches taste slightly sweet is that there's a gene for amylase, which is ordinarily expressed only in the pancreas in most mammals, but in humans the gene has been duplicated, and then a retrovirus with a salivary gland-specific promoter sat down next to it, and that causes this gene now to be expressed in the salivary gland as well. And that gives us, we would argue, a penchant for eating starchy foods as opposed to simply foods that are naturally sweet. And they have a lot to do with our originally settling down to become farmers, growing wheat and things rather than just gathering fruits and nuts from the trees.
So this is these kinds of effects are from infection, but the endogenous sequences can also move around in the genome. Is that correct?
In a sense, yes. Once a provirus is there, it's there. It doesn't move. The provirus doesn't move.
It can duplicate,
but it can be duplicated.
Okay, so it can make some RNA, which can be reverse transcribed and then go jump somewhere else, right?
But with genes that come from, with endogenous retroviruses that come from viruses, for the most part, that kind of purely intracellular event is rare.
Okay.
The process of retrovirus replication has a requirement, really, for release of the virion from the cell and infection of the next cell. Now, some endogenous viruses, a few, particularly, there's a couple of elements that have lost the ability to do that, and now do in fact retrotranspose just intracellularly.
Right.
But that's a rare exception. Most endogenous retroviruses, as far as we can tell, still require that they be expressed as a virus, come back. They actually don't leave and come back from the same cell. They're probably from one of the other 10 to the 14th cells or whatever in the body, and make their way back into the germline.
Right, if they're able to do that, because many of the So though the human endogenous retroviruses are dead, they can't make particles, right?
Right. Most of them are dead. This is actually a much more recent part of our research is looking at human endogenous retroviruses, and most human endogenous retroviruses are, in fact, all of them are not. Well, let me put it another way. None of them are completely competent for replication.
Mm-hmm. Mm-hmm.
A few clever people have taken the viruses of the most recent group, which is a virus called human endogenous retrovirus K, or HERV-K,
right.
And which we know there are some members that were integrated fairly recently, that is to say within the past few 100,000 years.
Mm-hmm. In our ancestors, right?
In our ancestors, but still,
not Homo sapiens,
more or less,
but
approaching human ancestors a few 100,000 years ago. And maybe there's still some around today. We just don't know that.
Mm-hmm.
We don't have a good way of telling. We're trying to figure out how to learn that in the lab, but we haven't gotten there yet. And but there are about 20 or so different proviruses, more or less. It varies a little from one individual to another, in the human genome of this age that clearly come back from a common ancestor about this time.
Right.
And so what these people do was logicked. There were two labs that did this. One was here in New York, the lab of Paul Bienoche. The other is in France, Thierry Heidman. And what they did was they reasoned that the ancestor of all of these must have been an infectious virus that integrated independently into these different sites. In the course of that integration, or subsequently, they've all suffered some mutations.
Yeah.
Independently, that rendered each one infectious. But if you could infer what the common ancestor was by looking at the sequence of all these and reassemble that common ancestor from the bits and pieces that you have now, you might get something that was infectious. And indeed, they were able to do that. They were able to sort of resurrect something like what the original infecting virus was. It probably still has some defects, so it doesn't replicate extremely well, but it does replicate. And so show that this is and then learn something about the properties of these viruses.
So when we say we have 8% of our genome is retroviral, this is what we're talking about, mostly the proviruses. These
and lots of other things that
pieces,
most of which are much older.
Okay.
Yeah, the most common thing that you see in the human genome, with this group of viruses, for example, altogether, I said there are about 20 recent ones, but there are a number of much older ones. Altogether, you can count to about 90 of them in an average human genome, varying a little bit from one person to another. But if one of the common things that happens to proviruses is that they, because they have this 1,000 base or so long terminal repeat at either end, those can occasionally base pair with one another, and you can get a recombination event, a crossover between them, that has the effect of popping most of the viral genome out of the chromosome and leaving behind just this solo LTR. And for this particular group of viruses, as I said, we have about 90 intact viruses, but there are about 10 times that many solo LTRs. So most of the ones that ever came in have gone away in this way. So most of the viral DNA sequences are solo LTRs or other kinds of fragments. Very few of them. There are a number of other virus groups that are represented by more or less complete proviruses, but they're all much older than the HERV-K group.
So many people are always interested in why we have kept these sequences for so many years. I know at least one gene is syncytin, which is a placental gene, right? But what about all the other retroviral sequences? Why are they still there? Do we have?
Well, I already alluded to one other thing that they do probably. And that is offer protection against infection with the same or closely related virus from outside. And that may have been a selective force for a lot of them. If they, in fact, contribute to the extinction of that virus from the wild, as you were, from being an exogenously transmitted agent, their job, in a sense, is done. But they'll hang around for a long time afterwards. It takes a long time to get rid of sequences from the genome. So these sequences are hard to get rid of.
Okay. So they may just
and there's probably no strong drive to get rid of them. But it's interesting. Some groups have shown, for example, not us but others, if you sort of estimate the age of proviruses and you look at where they're located in the genome with respect to their age. If you look at very recent ones, they can be anywhere. They can be in genes, and we've identified in the course of our work with endogenous mouse viruses, MLVs. We've identified a couple that were associated with very distinct mouse mutations. One particularly interesting one was a gene called hairless. And that mouse is exactly what you think. It has no hair.
Right.
It's not a nude mouse. That's a different mutation. And it was originally discovered in an aviary in England, where the night watchman was, sort of dozing off one night and looked up and saw this very strange-looking pink mouse running across. And I guess he was something of a mouse fancier, so he caught it and brought it back and brought it home and bred it. And eventually the mutation called HRS was bred into was crossed back into inbred mice. And in the course of mapping MLV proviruses, which Jonathan Stoy and Wayne Frankel did when they were postdocs in my lab, we discovered that there was an endogenous provirus that we hadn't seen before. Destroying that. That was very tightly linked to this hairless mutation. We could not find a mouse from any cross involving this where those two had been genetically separated. Now that doesn't place it really close, and genetic crosses is the closest you can get is about 1 million base pairs.
Yeah. So we didn't sequence this and figure out where it was, right?
No, we figured it out differently.
Okay.
We figured it out in a different way, and that is, it was known from previous work by another group that with another mouse mutation that also caused a code color change in this particular case, that if the provirus went away but left behind a solo LTR, which is, as I said, is a fairly common thing, then that solo LTR was no longer caused the phenotypically visible mutation. The coat color reverted to a wild type sort of dark brown coat color of mice. And so we reasoned that maybe the same thing was going on here. Well, a strain of hairless mice, as it turned out, you need to breed large numbers of animals. This is a fairly rare event, this kind of LTR recombination. So, but what Jonathan did was to start asking around, and it turns out this hairless mice of this strain are used a lot in Australia, and they're used for testing sunscreens. It's kind of mouse is a good model for testing cosmetics and things like that, as far as their ability to either protect the skin or to irritate the skin. But of course, with all that hair, it makes it a little hard. But if you have a mouse that naturally has no hair but is otherwise fairly healthy, then that's very convenient. So they actually bred this mouse in the U.S. They were always bred as heterozygotes, which are wild type, which have hair. Yeah. It's only the homozygous animal that has the mutation on both chromosomes that doesn't. But they were being bred as homozygotes in Australia because it's much less wasteful if you need to breed these animals in large quantity. And sure enough, every now and again, a mouse would show up that had two parents without hair. Then now this mouse is one of the litter had hair again. And so he found, Jonathan got a hold of somebody there that had some of these mice, looked at them with the hybridization probes, no PCR at the time, with hybridization probes at the time specific for this provirus, and was able to demonstrate that the provirus had gone away, leaving behind a solo LTR right in the middle. And that really allowed him to both identify and eventually to clone the gene for hairless.
Interesting.
The interesting follow-up to that story actually happened right here in Columbia. There was a group that was studying a condition called alopecia universalis, which is a total loss of body hair.
Right.
Like these mice, the individuals are born with sort of normal amounts of baby hair. That's true of the mice too. The hair grows in initially, but then falls out almost right away. And it turned out that, lo and behold, that's caused by a point mutation, that they were able, using the information that we'd obtained from mice, they were able to show that this was a point mutation, not a virus, but it was in the identical gene.
Okay, not driven by a proviral mutation.
Not driven by provirus. The same thing is true with lots of cancer.
Yeah.
The genes that were originally shown to be responsible, that were originally shown to be driven by provirus insertion in mice or in chickens to cause cancer, where many of the same genes were subsequently shown to be very important in human cancers.
Right.
The MYC in the chicken genes, for example, was one of those. It's very important in human cancers. RAS is another one. It's 50% or something of human cancers are probably associated with mutations in RAS, not involving viruses.
Right. So does every animal on Earth have endogenous proviruses?
Certainly every mammal.
Every mammal.
And probably every vertebrate. Mm-hmm. And you can find it. There are many invertebrates, flies, mollusks. Mm-hmm. In fact, Steve right next door here,
steamer clams,
has been working on a virus. They call an element they called steamer.
Yeah.
Which is more or less a virus, a retroelement anyway.
Do they have particles? I don't know.
I think there are particles, yes.
Okay.
This
is in a steamer clam, right?
Yeah.
We have to get him to talk about this story.
Yeah, you should get him to talk about it. Yeah. There's some interesting follow-up to that story.
How about, so fish do, right?
Fish as well. Probably every vertebrate. Certainly every mammal.
Some insects,
probably every vertebrate. At least some insects. And mollusks. So they've been around a long time. I'm sure if you got a block of amber with a mosquito in it, you could get some dinosaur blood out and find endogenous viruses in the dinosaurs.
Oh, I'm sure that wasn't that long ago, right? So were the endogenous virus sequences before retroviruses or after? They're always
maybe there's some sequences that predated retroviruses that became retroviruses, but they're lost to us, I think.
Okay.
I would be very surprised if anything visible turned up. Everything that we know was a retrovirus that then went back into the genome.
So mice have endogenous retroviruses. Some of those make particles, right?
Some of them do. Some of them make infectious virus.
But humans do not make infectious viruses.
Humans don't make infectious viruses. Some of the HERV case proviruses do make particles. In fact, normal human placenta has...
Really?
You can always see particles in normal human placenta. So there's some kind of... at least one of those proviruses. Yeah. It's probably only one of the 100 or so proviruses is doing that. But at least one of those proviruses has a promoter that is responsive to signals in the placenta. Human cancers very often make... Breast cancer, for example... Really? Very often makes large amounts of this virus. Germ cell tumors, particularly teratocarcinomas, make even more. They really make a lot of this. They really pour this out. I think...
But they're not infectious particles.
But they're not, in no case are these infectious particles. Every single one of these proviruses is, in fact, defective in some way or another.
All right, so chickens make infectious. Some chickens make infectious. Are there other species that also...
Well, mice and cats.
Mice, cats also.
Pigs also.
Okay, and the pigs are a problem because of transplantation, right? We're worried about PERVs.
There's been a lot of concern about that. So far there's been... Well, it's actually quite interesting that of the 4 closest companion animals to humans, 3 of them have infectious endogenous viruses that they express.
That's cats?
Cats, pigs, mice. Dogs would be the 4th.
Do they make viruses too, dogs?
Dogs don't.
They do not, but they have endogenous viruses.
They have endogenous, but they're all like human. Yeah. As with humans, they're all quite old and decrepit, as it were. But as far as we can tell now, with the XMRV scare a few years ago, there's been a lot of work looking for evidence of infection of humans with these viruses, and there was a lot of work looking at these also back when feline leukemia virus was first discovered back in the 70s. Because of the concern there might be cross-species transmission into humans of these viruses. So far, there's no evidence that a single person has ever been infected with these viruses. And
including XMRV, right?
Including XMRV. Human cells were infected with XMRV. Right. Many human cell lines have been infected with XMRV.
Yes. But no humans.
But no humans. And if you try to infect human PBMCs directly out of a person, peripheral blood mononuclear cells or blood cells, the infection dies very quickly. And if you inject large amounts of this virus into a rhesus monkey, we did this experiment in Frederick, I was a co-author in the paper, but other people actually did the experiment. The virus infection dies very quickly. What gets left behind are proviruses that are very heavily mutated. And in my opinion, the mutation is caused by what was a protein that was originally identified as an HIV restriction factor called APOBEC, APOBEC 3G or 3A or 3F. There are a bunch of different forms, but those two are the most important ones. And I think those are have been very important in protecting us against infection.
These enzymes get incorporated into the particle, and then they mutate the genome, right?
Right. And so you can get a little blip of infection when the virus first comes in, but then it dies very quickly, and all it gets left is this very heavily mutated provirus.
Because these viruses don't have the antagonistic proteins.
Not against the human. They have something that protects them against the APOBEC in their own species. We don't know what that is, actually, in most cases. But we don't, and that protection is not perfect, because we know that mouse APOBEC also it can affect the replication of some mouse viruses.
So since you mentioned XMRV, I wonder if we could just sort of review the entire story. So it was first discovered as something in prostate tumors.
It was first discovered by looking for a virus in prostate cancers, particularly because there's a mutation that gives rise to an enhanced incidence of prostate cancer in certain families. So that mutation was traced to a gene called RNase L, which is known to be part of an antiviral response that's mediated by interferon. It's interferon-induced. Probably very important, actually, in poliovirus, for example. And so that led to the hypothesis that because this mutation inactivates this gene, and these people then have a higher propensity to prostate cancer, maybe that means there's a virus involved in at least some cases of prostate cancer. So they went looking, and from that point on, it becomes it's sort of like they say about plane crashes. So lots of things have to go wrong in order, and so lots of things actually went. So a bunch of different things actually went wrong. Because now we know.
You can tell us what happened.
The first thing that happened was an analysis was done by the geneticists who were working on this. They went to some people who had what's called a virochip. You've probably talked about that on TWiV a few times. Which is a microarray that has lots of virus sequences on it. You can use that. You can take some sample from a person suspected of being infected with some virus, although you have no idea what. You can then sort of randomly PCR amplify everything that's there, and then you can hybridize it, put it onto this chip, and look for a signal that in a well it corresponds to this virus or that virus. And when that happened, the only signal they got out was from murine leukemia viruses. Okay. The reason they saw that, to sort of fast track ahead for a minute, was probably because of a general problem. Actually, this goes back to when we met before in Boston, that mouse DNA is everywhere.
Right.
In particular, there are some reagents like enzymes that are used for PCR amplification, certain strains of TAC, that are modified to be what they call hot start. That is to say, they're inactive in the form that you buy them. You have to basically boil them in order to activate them to start working, and that helps to enhance the specificity of PCR reactions. Well, one of the ways in which some companies make TAC inactive that way is to complex it with a mouse monoclonal antibody against that
the TAC protein.
Against that protein, and that inactivates it.
Yeah.
Well, the mouse monoclonal antibody is grown often in mice. I don't know if it is in this particular case, or in mouse cells in any case. It's inevitable that if you're looking with enough sensitivity, there's going to be some mouse DNA in that preparation as well. And when you start PCR amplifying kind of randomly, a mouse cell has about 100 copies of DNA that would react with these murine leukemia virus probes. So it's there's a lot of sequences in there, and so that was probably, we don't know this for an absolute fact, but that was probably the source of that signal that was originally detected. So they went back from there, and then they tried to find a virus, and lo and behold, they started to grow out this virus, which they named XMRV for xenotropic MLV-related virus, horrible acronym. And they then found this virus in some cases of prostate cancer, and some other labs started finding evidence for it, for the same virus. Some labs here in New York, for example.
Right.
In fact, I think even here at Columbia.
Probably
at the time.
Yeah.
And started finding it also in prostate cancer, but it still stayed at a fairly low level until another group, in collaboration with the first one, found what they claimed was strong evidence for this virus and antibodies against this virus and isolation of this virus from cases of chronic fatigue syndrome.
Right.
And that really caused a huge stir. That was a huge flap came out of this, because chronic fatigue syndrome is one of these very mysterious conditions. There's no good biomarker for the disease. There's a very strong patient advocacy group, and many of whom feel that, probably justifiably in many cases, that they have been sort of mistreated by the scientific establishment in terms of effort to help by finding a cause or potential treatment for this condition, and which suffers again from the problem that there's no really good biomarker for the disease. You can't do a CD4 count like you could for AIDS and show that the CD4 cells had gone away for a clear marker for the disease. There's all of the analyses that you do are really quite vague, and you put them all together and you get a diagnosis, but you do that also by excluding other things.
Right.
And so it's a very messy situation, and if I were suffering from that, I would feel quite put upon too, actually, I must say. But having a cause of this disease, even without, in the first place, gave rise to the possibility there may be some treatment, as there was, for example, for AIDS. But in the second place, sort of validated the existence of the disease and the seriousness of the disease in the eyes of the patient community, some of whom are quite vocal, as we learned, very outspoken. Again, like analogous to the AIDS community back in the early days. And so this created a big stir. There's a lot of research was done. A lot of people who had worked on viruses, murine leukemia viruses, which this clearly was one of, really came back, reappeared all of a sudden.
What point did you get involved with it? Early on? I
got involved pretty early on because, partly because of the NCI connection. Some of the...
it's Lombardi, right?
Well, Lombardi was out in Nevada.
I'm sorry, the guy, fellow Ruscetti. Frank Ruscetti.
Frank Ruscetti. Frank and Sandy, both of the Ruscettis at NCI.
Okay.
And so it became, and there was a lot of retrovirology expertise in the NCI, and a lot of people who'd worked on murine leukemia virus in past years. So there's a lot of expertise that we could bring to bear on this virus quite rapidly. And so we started to work on it. A lot of groups there, including me, started to work on it for a while. And we'd also been working, my lab in Boston, we'd been working for a long time on endogenous murine leukemia virus. And I had a student there who was particularly interested in this. The virus had a somewhat unusual property, particularly if you think about HIV, and that is that different isolates from different patients actually were very, very similar in sequence. They differed a little bit, but the similarities were much more striking than the differences. I had a very nice hypothesis, which was just wonderful, that would explain that. And that is that this really was an endogenous virus of mice. It was very closely related. It was not identical to any of the known endogenous sequences we had in mice. And we'd looked at a lot of them. We'd looked at hundreds of them. But it wasn't identical to any of them. But
yeah, I remember you showing a slide in Boston with all the endogenous sequences you had. You said none of these matched.
None of them matched.
Yeah.
So I set a student to trying to see if she could find it, because the hypothesis was that yes, it was an endogenous virus of mice. It was out there somewhere in wild mice, not necessarily in the inbred lines that we were looking at in the lab, and that analogous to, say, the hantaviruses, like Four Corners viruses, that occasionally when you get a bloom of mice, you get a bloom of this virus and you get a lot of people infected. The same kind of thing might have been happening here with starting with an endogenous virus of mice, which, because it was in the germline of mice, changed very, very slowly. So you could have essentially the identical virus appearing in outbreaks in completely separated, widely separated parts of the country, and still have exactly the same sequence. So I thought that was a wonderful hypothesis to explain that. So I set away my student to working on this, and after actually a heroic job, she managed to find a sequence that was identical to the virus, but it was only identical to about half the virus. And it was about 10% different from the rest of it.
And she found this in a strain of mouse somewhere?
She found this in a strain of inbred mice.
That you hadn't looked at before?
That we hadn't looked at before. Right. Exactly right. We had looked at a bunch of them. This one we hadn't happened to have looked at.
Okay.
So she went through a lot of them, and she found it in this one. At the same time, some colleagues of mine down in Frederick were taking a somewhat different approach, for which I need to back up a little bit. So shortly after this XMRV association was made with chronic fatigue syndrome, it was discovered, and prostate cancer, it was discovered that there was a prostate cancer cell line called 22RV1, which had been derived from a prostate cancer originally. And when a group out in Seattle looked at the cell line, they found that the cell line was making substantial quantities of virus, which again proved to be genetically essentially identical to XMRV. So the hypothesis then was, fine, this is a prostate cancer that in fact was caused by infection of this virus in the first place. Well, as it happened, my colleague, Vinay Pathak at NCI Frederick in the DRP, and his wife, Wei Su Hu, also has a lab right there, had access to this cell line. Let me back up a little bit. The cell line had been generated by passage of this prostate cancer through nude mice, right, which was a very common way, still is actually, a common way to both study human cancers and also to adapt them to cell culture. Human cancers are very difficult to get into cell culture. Very few of them will go directly into cell culture without sort of previously conditioning them and selecting for some variants that can grow better by passage through some sort of immunocompromised animal. A nude mouse is the preferred one. So he was able to get a hold of some DNA samples that came from passages of this prostate cancer through nude mice, because his wife's brother-in-law, his wife's sister's husband had actually worked with this cell line, had actually worked on these cancers some years before at Case Western Reserve, where they'd been isolated, where this has been isolated. So they happened to have these old DNA samples in the freezer. And so he went back and looked, and so from those DNA samples, he could go looking for this particular provirus by simple PCR tests, and he found that in samples from fairly recent passages, which the cell line was the tumor was passed through mice between about roughly 1990 and 2000. After 2000, it was taken into cell culture and it was grown in cell culture for another 10 years before anybody realized it was making all this virus.
Right.
And so it was passed around the world. There are about 300 papers, none of which recognized from many, many labs worldwide using the cell line, none of which knew that this cell line was making like 10 to the 9th particles per milliliter or more. And, of course, the potential then for cross-contamination of other cells in the lab and so on is very high. This is not the only case of this.
And by the way, when this was found out, a lot of people said, this is horrible that we're seeding the world with this virus. Who knows its pathogenic potential? But you said in non-human primates it doesn't do anything, right?
In non-human primates it dies very quickly.
And in explanted human lymphocytes it doesn't do anything.
It dies very quickly.
All right. So the pathogenic potential for people is zero probably, right?
Right. It's extremely low, probably nothing.
Okay.
Fortunately, which is a damn good thing, actually, I must say. So he went looking for this virus in these DNA samples from the earlier passages of this, and he found them in the recent ones. He could find this virus present in basically all the DNA. Every cell seemed to have this provirus in it, a provirus for this virus. But when he got back to older ones, he couldn't find this. He couldn't find that signal anymore. So the virus was not there in that cell line when it was first put into mice. But if he looked really hard, he could find a signal in the DNA at a much lower level. And that signal was not almost certainly coming from the human DNA, but was coming from mouse DNA. There was also, these were tumor samples from mice. They inevitably had some mouse DNA in them.
Right.
And what he found was a sequence that corresponded exactly to half the provirus. So I still remember the day very well when he and I were sitting in my office down in Maryland. And he said, well, I'll show you mine if you show me yours. And we put these two sequences together, and they fit like two hand in glove. You could just draw a series of about 5 crossovers between those two and get exactly XMRV back out.
Right.
Leaving really no doubt. And then we were able to go back to mice of the strain that were the nude mouse strain that was used for these, and we were able to isolate both of the individual proviruses and show exactly how that recombination event must have occurred. So what must have occurred is that these prostate cancer cells make a very good host for this virus.
Right.
And this virus that eventually arose was a recombinant between these two proviruses, which we called pre-XMRV1 and pre-XMRV2, that were present in the nude mice. And they're present we can still find these proviruses in nude mice today, although they are not present. Nude mice are outbred and they're not present in every single one, but a lot of them have both of these proviruses. Right. And they're probably the only strain actually of mice that has both these proviruses. It turns out. So it's a whole bunch of things had to go wrong at the same time.
So now we know this virus is an artificially generated virus. So how did it get into the original prostate study? So which was done in San Francisco, right?
So that was just a signal on the viral chip, and that was probably caused by mouse DNA that was in the enzyme that was used to make the.
But they also sequenced something, and they got XMRV out of that.
They didn't. That was the virus that was sequenced later on. So this study had been done at Case Western Reserve. Well, it turned out that's exactly where this prostate cancer cell line had been generated. So that it was probably a cross-contamination in a liquid nitrogen freezer or something like that.
And
most likely.
And how did this virus get into the chronic fatigue study?
Well, that was done in collaboration with a group at Case.
Okay.
And so samples were passed back and forth between those two groups.
All right. So the whole thing was a contamination, basically, and the virus was generated. It's a mouse
several
virus,
different kinds of contamination, and then there was another study that was done that was also telling by Lowe and Alder and people that where they looked at samples that had originally come from a chronic fatigue syndrome study in Boston,
right?
And by again some very sensitive PCR managed to identify small fragments of genomes that looked like endogenous murine leukemia viruses. They were not exactly the same sequences as MLV. In fact, they were almost exactly the same sequences of endogenous murine leukemia viruses that we had previously identified. And if you did a phylogenetic analysis, they all fit really very nicely together. And control DNAs that they looked at did not have these sequences in them. However, there's a very important lesson there, and that is that controls need to be controls. So the samples that were looked at were from the study that was in the freezer up at Harvard for many years. The controls were actually taken from discarded blood bank samples, blood samples that were around at NIH. Often in cases like that, and the same thing was probably true in a lot of the chronic fatigue syndrome samples. What happens is that people go in and out of the samples a lot. The controls are much less handled, so the samples get handled a lot more. As I say, mouse DNA is everywhere. There are lots of potential sources. I visualize, for example, a big storage shed somewhere that's got a big pile of, let's say, sodium phosphate. And every once in a while, somebody goes in with a stainless steel shovel and grabs a few kilos of this, and that gets made and gets taken over and made into your reagent grade phosphate buffered saline. Yeah. Well, that's what happens during the day. At night, there might be mice crawling all over this pile, leaving traces of their DNA and whatever else behind that could eventually get into some of your reagents, but not others. I was a co-author on a study with my colleague Bridget Huber up at Tufts during this time, in which it turned out that many of the controls were positive for mouse DNA, for MLV DNA, but not the chronic fatigue samples, which is just again because they were not in the same tubes, they were not handled exactly the same. So eventually, the study, when Ian Lipkin did his study, his very expensive but fairly definitive study to exclude that ended up excluding any evidence whatsoever of XMRV infection from human populations, particularly from chronic fatigue syndrome. They were very careful to centrally buy all of the tubes, the reagents, and everything else that was used, distribute those so everybody, controls and cases would all have exactly the same, be handled exactly the same way with exactly the same reagents and exactly the same tubes and exactly the same syringes and everything else. And I think that was critical to avoiding this kind of problem.
I think it really underscored how much mouse DNA. Actually, there's Eric from San Francisco. Delwart says DNA is everywhere.
Yep.
All kinds of DNA.
Yep.
He said it's a real problem.
Yep.
And this underscored that. And then you combine it with a very vocal patient group that would like to find a cause for their illness, and some zealous investigators who think they've found the cause, and the story was really remarkable and had a life of its own, but now has died down, and unfortunately there's nothing else on the horizon for chronic fatigue syndrome.
One of the investigators who's most deeply involved in this, though, has a book coming out very soon.
Oh yeah, it has been published, I think.
I don't think it's out yet. I think they kept postponing it. But
are you talking about what's her name?
Judy.
Her book.
Yeah.
Oh, it might have been postponed. Yeah. It's Well, I'm sure it'll be a bestseller. Be interesting.
I will eagerly await your review.
You're not going to read it. It's funny. We covered this a lot on my blog on TWiV. My wife doesn't follow any of this stuff, right? She's a scientist, but she doesn't. I don't know why, but for the past few years, we have a high school son who's been ill, and she suspected chronic fatigue for a while. And she came across one of the XMRV papers, and she sent it to me. She said, "Why don't you check for this virus?" And I said, "Don't worry, we don't have to."
Don't worry.
We don't have to check. I didn't explain because, it was a long story.
It's a very long story.
And, there were some really remarkable follow-up papers where I think Charles Chu, among others, traced by mitochondrial typing which there was a little bit of mitochondrial DNA left in some of the samples, and he could say, yes, this definitely came from contamination from these virus-producing cell lines and so forth. Really an amazing amount of work. I mean, the science paper you published was remarkable. that you could identify this. It's beautiful science. Unfortunately, you would rather it go the other way, that it identified something
rather than de-identifying. Yeah, exactly. Yes, I completely agree with that.
But it's totally beautiful. So it's good to hear the end of that. I want to just talk a little bit about what you talked about today. You have two jobs. You have one at Tufts and one at the National Cancer Institute. And you're very happy to do that? You go back and forth?
I've been doing this for a long time, since 1997.
And you don't mind a few days each week at each place?
I've gotten quite used to it, actually.
And you fly back and forth?
Fly back and forth.
Huh. My wife in December started working 3 days a week in North Carolina. And she goes down there
kind of thing.
And then comes back here. But she's hoping it won't be as long as yours, though, anyway.
I find North Carolina more annoying to get to because the thunderstorms there in the summer can be horrendous.
Mm-hmm. Yeah, the flights are always canceled or delayed. And it's not... you fly into Dulles?
National, usually.
So you can probably have more flight options from Boston.
There's a lot of options.
There's not a lot to Raleigh-Durham from Newark, surprisingly. Anyway, you went there, as you said today, to work on AIDS and drug resistance, right? And so you said a couple of things that I think are really interesting. You showed a beautiful slide, by the way, of the effect of giving triple antiretroviral therapy to an AIDS person. There's a big decline in virus loads.
Exactly.
Is that published, that image?
Oh, yeah. That particular one, I don't know. It might be in a review somewhere.
Because you had someone's name on it.
Yeah, it was drawn. It was based on data that we and my colleague John Mellors and people at NCI had obtained.
Bill Hildorfer.
You're right. Good catch.
I just copied it down because I wanted to ask you, because I'd love to use it.
He's, I believe, a student or postdoc in John Mellors' lab at the University of Pittsburgh.
Love to have that. Anyway, so the loads get very low.
And there's a decline that can be broken into phases.
Right.
And it's important to remember that the virus load, the level of viremia in an HIV-infected patient, is most important because it's a fairly immediate measure of the number of productively infected cells, at least that are releasing virus into the bloodstream. Because the virus, once it's released, it turns over very rapidly. So, so that as the number of cells declines, the amount of virus that are producing virus declines. The virus level of virus declines very.
So
along with
before antiretroviral treatment, what percent of CD4 cells are infected?
It's about more or less 1%. 1%, let's say.
And then it goes far below 1%.
And then, as far as productively infected, it clearly goes far below. It goes down by at least 4 orders of magnitude, down to the lowest that we can detect. If we use a very sensitive assay that we developed, this is one of the reasons I took the part-time job at Frederick, was to be able to develop assays like this. If you use the very sensitive assay that was developed down there, we can detect virus. We can detect virus down to less than 1 copy per milliliter. Less than 1 copy of RNA, which would be half a virion per milliliter of blood. So it's very, very. The assay is very sensitive. In the average patient, it starts off at about 30,000. So it's more than 4 orders of magnitude decline in most patients.
All right, so there is clearly a little bit of virus in the blood, even on triple antiretroviral.
A little remains, yes.
And this turns over and it's produced again by some latently infected cells, but very, very small amount.
Very small amount. It's very hard to identify.
And it's not transmissible, right? If these individuals
these individuals do not efficiently do not really transmit HIV. On therapy of the infected individual is a good way for preventing infection. In fact,
they can have unprotected
sex. There's a so-called test and treat. Well,
I don't know.
The answer is yes and no, because they're not being monitored constantly. I suppose if they were to take a viral load test immediately at this time, but the problem is that if you miss a few pills, the virus can actually come back again. Sometimes you can bring that back under control. Sometimes you'll get resistance, and that you can't control it again, at least not with the same drug regimen.
Yeah.
So you can never be absolutely sure that that hasn't happened. So unprotected sex is not a good idea.
Okay.
Even for people on suppressive therapy, even if they think they're very well suppressed.
So you showed on a slide, if you stop taking a pill, the virus goes way up again. It shoots up really quickly.
No matter what, even after decades of therapy.
So the viruses then, what's happening is the few viruses that are present are able to now infect new cells in the absence of the drug, and they multiply very quickly.
Exactly.
And you have studied those, and you find that there's very little genetic variation, both during treatment and after treatment.
Well, there's very little genetic distance. The virus that's around before treatment, if you look at the genetics of that population, you find that every genome is different in patients that have been infected for a long time. And if you were to look at an untreated patient with time, you would see evolution of the virus sequences. So you see accumulating genetic distance by accumulating numbers of mutations with the years after after infection. However, if you treat a patient and put them on suppressive therapy, you find that the virus that you look at has stopped accumulating genetic distance. It just does not evolve any further.
Okay.
And in fact, it devolves in a sense that you start to see accumulation of clonal sequences, identical sequences, which are not present initially, that reflect outgrowth of specific clones of infected cells in the patient, as it turns out. And so the virus complexity, in fact, goes down with time after infection. It doesn't increase the way it does in an untreated individual.
You said something that's really remarkable. You said antiretroviral therapy for AIDS is the best we're going to get. It works really well.
As far as its ability to block virus infection, yes.
So if we could
I don't think we can do any better. I don't think we can improve that to suppress the virus any better than we. And we did experiments. I didn't talk about this for lack of time, but we and others have done what are called intensification experiments, taking patients taking 3 drugs that have this very low level of viremia, which we can detect with these sensitive assays, and then adding another drug to those patients of a completely different type of drug. If the virus, if any of that virus that we were seeing were due to replication, you would expect it to go down still more with a new drug, and that never happens. It always remains very constant, and then you take that drug away, and it doesn't go up either. It just stays exactly the same, and that's what I came to call a YANIS study, YANIS, which stands for yet another negative intensification study.
So if we could get a triple pill to everybody who's infected, which is impossible, of course, we could get rid of the reservoir. Well, you could suppress the replication, but you'd still have latent genomes in these individuals, and that's the problem, right?
Right. And which is not a problem as long as they keep taking drugs, and as long as they keep taking drugs with sufficient regularity that they don't allow
transmission.
They don't allow evolution of resistance, is what I was going to say, or transmission for that matter.
Yeah. So if you
But in fact, the so-called test and treat strategy is another approach that is being used in a public health sense for HIV to identify couples where one individual is infected and the other one not, and to make sure the one that's infected really is well treated to suppress that, so to prevent spread of infection to the partner.
If you take a triple therapy every day religiously without stopping, will you eventually evolve resistance?
No,
you will not. So
as long as the therapy, as long as it's taken uninterrupted, and as long as there was no resistance to begin with, occasionally individuals are infected with a virus that had come from somebody who had failed therapy by resistance, and so you get transmission of the resistant virus. That happens. That's why now in the U.S. standard of care is to actually determine the sequence of the virus that's in a person before you start therapy to make sure that hasn't happened.
But that doesn't happen everywhere, that standard of care, right?
No, that doesn't happen. That certainly doesn't happen in Africa.
Yeah, okay, got it. So Yeah, that's it. So whenever there's a drug failure, it's because you're either interrupting, you're stopping, or you were infected with a resistant virus and you didn't detect it to begin with.
Right.
Okay. So
I should say once in a while, the reason the drugs work like that is that mutations to resistance don't arise after you start therapy. They're already there in the virus population. If they're not already there, they're not going to appear if you really stop replication. There's no way for them to appear. Of course, yeah. And that for a single drug, there will always be a fair number of mutations. Very easy to model it. There will always be some mutations already present in the population that give rise to resistance. Even for 2 drugs, there will be some that are resistant to both. But when you get down to 3, the frequency of those resistant mutations will be so low that almost nobody will have a virus in their population that is already resistant to those 3. Now, every once in a while, there may be somebody that's so unlucky that there is. That actually has appeared, and you may get occasional failures for that reason. So, fortunately, now in the U.S., Northern Europe, Japan, and other fairly wealthy countries, we have quite a few drugs available. So, if you do get this kind of failure, or if you get intolerance to the single pill, triple drug regimen, which is not terribly uncommon, then there are things you can switch to.
Right. So, do you think it will be possible to eradicate infection using triple therapy? Because Tony Fauci seems to think there's good chance.
In a population sense, that's a possibility. Yes, yeah. It's a possibility, but it's a very long-term prospect. It just takes a long, long time, and you have to keep it up, and you have to keep it up in a population basis in populations that are sometimes, in the U.S., the populations that are infected with HIV are ones that tend to be very hard to reach.
Okay.
They tend to be, the concentrations tend to be in very poor, poor communities, and tend to be in IV drug abusers and other groups that are difficult to do this way.
You talked. I don't know if you want to talk about this. You let me know. You talked today about integration sites and clonal expansion. Is it all right if I ask you a little bit about that?
Most of that's been published, actually.
So you've done a study where you look at integration sites in patients who are on treatment where HIV is going in the genome. And normally the integration site is more or less random, right? There are some hot spots. Right. But you find enrichment of particular sites.
Yes.
And why is that?
Well, there are 2 reasons. The simpler reason is that the provirus is simply marking a cell that was chosen to be expanded for other reasons. We're talking about T cells that are part of the immune response that are antigen-specific T cells. If that particular antigen, that the T cell that this provirus is sitting in happens to, if that antigen happens to present itself, this T cell will be expanded, and the provirus will then mark that, is simply a marker for that specific cell. Right. So that's one thing. And we think that's true we think in a number of cases. But in other cases, the evidence is quite compelling that the virus itself is, in the integrated provirus, the DNA form of the virus, is in some way participating in the process of expansion, of growth of this, or survival of this particular cell, and its daughters and granddaughters and so on. And the evidence for that is that although integration is more or less highly distributed across the genome, in this particular case, many of the integrations, a very, very high fraction of the integrations we look at, tend to be concentrated in a very small, in different cells. None of these are in the same cell. Different cells tend to be concentrated in a very, very small portion of one gene or another. And these genes are ones that have been identified as being important for the control of cell growth and division differentiation, particularly lymphocytes, B cells or T cells.
They're not oncogenes, right?
They have been. One of them has been identified as what I would call a real oncogene, because as a virologist, I think a real oncogene is only one that's been identified by an experiment, by a virus experiment.
Right.
And so one of them actually has been in mice, has been identified as as an oncogene that is activated. Quote, it's a strange term when it's as though it's as though the real function of this gene was to cause cancer.
Yeah, right,
it's
right.
not.
So in retrospect, the name isn't great anymore.
Abused. I like it. It's been abused by integration of murine leukemia virus in a mouse experiment to cause a B-cell cancer. And that was shown about 5 years ago by a group in Sweden. Denmark, I think. And if you're an old-time retrovirologist like me, what you see looks very much like what you see in retrovirus-induced cancers in chickens and mice. That is to say, you see amplified integration of a provirus in a position where it's likely to affect the expression of the gene that it's in. And that's the case. We see a remarkably high concentration of integrations, many of them amplified by cell division in a very small portion of the genome, a region of about 5 kb or so, 6 kb, which is 1/1 millionths of the total cell genome, has attracted about 10% of the integration events. It's really remarkable, and all of them pointing in the same direction. And anybody that works on retroviral DNA integration knows that the integration system just doesn't know anything about transcription. It doesn't know which direction transcription is going in. It has no way of being directionally specific. Just the fact that all of these are 15 or 16 different sites are all pointing in the same direction itself is a very, very high probability of being meaningful. The probability of that happening by chance is very, very small.
So HIV is associated with cancers, right? Later in
HIV is associated with cancers.
So are any of these causative?
I think we think that's a possibility. If you ask many AIDS physicians, they'll tell you that's just doesn't happen. But we think there may be observational reasons why this hasn't. This may be, if this is happening, it might not have been seen because of the way people have been looking. Might not detect HIV proviral DNA even if it was present in some of these cancers.
So the way to look would be to take an HIV associated cancer, and sequence the integration site and see if it's in one of your.
We are in fact.
You're doing that?
We are in fact starting to do that.
It's quite interesting. I hadn't been aware of that area, and I think it's really, as you said, it harks back to Sark.
Yeah, exactly.
Where the virus goes in front of Sark and causes a tumor.
Right. And that's where tumor-causing retroviruses came from originally.
Right.
That's what happened to the original chicken that Peyton Rouse bought in the chicken market somewhere in lower Manhattan.
That's right. And he was at Rockefeller, and he injected it with a filtrate, caused more tumors, and eventually Howard Temin got a hold of that and figured out.
Peyton Rouse published that in 1911 and won the Nobel Prize in 1965.
Long incubation period. So let me end by asking you about cranberries.
Sure.
How did you get so Steve Goff introduced you as a gentleman farmer. You raised cranberries in Massachusetts. How did you get interested in that?
Well, a couple of things. One is as it happened when I moved to Massachusetts with my wife, back in the 70s, we bought a house across the street from a cranberry bog just by chance. It was just a nice setting. I mean, you don't have to look at a neighbor's house. You got this agricultural just out in front of you. So it was, it added to the sort of ambiance of the area. So that was one thing. And we watched them over the years, and we could see them take a lot of cranberries off this bog, and it never seemed to do very mumuch work there, which of course is an illusion. They're actually probably doing quite a bit of work at the time. And my next door neighbor, who was a banker and has a much better business sense than I do, came to the realization that this actually was a very good investment at the time. The price of cranberries was way down, but Ocean Spray in particular was developing a lot of new products, and clearly the market was building up and building up and building up. But the cranberry acreage was still relatively low priced. And so he thought this would be a very good investment. He and I, in fact, were both competitive gardeners. So we thought maybe there was something we could do with the skills other than just see who could grow the most unusual plant or the biggest rutabaga or something. And so we started looking around to see if there's a piece of property we could buy. And it took us a while, but because it had to be, they don't come onto the market all that often anyway. And it had to be sort of the right size and, within our budget and so on and so forth. And not too far away, although the piece we ended up buying was about 30 miles away. And
how many
after a while, we found
many
one.
acres is that?
72 acres of land.
That's big.
About 22 acres under cultivation. And
you
a
started
lot of wetlands, and
it was not a farm to begin with.
It was a cranberry operation. It was
okay, you bought it.
There were about 9 acres planted in cranberries. We bought it over the years when prices were good. We'd get a lot of money ahead, and we'd plow out some more of the acreage and build more cranberry bog.
I presume you hired
which is a
people
great
to
plan,
do this,
except
right?
for one thing, and that is everybody else is doing the same thing.
Is this where you grow cranberries in New England, basically?
Yeah.
Nowhere else in the country?
No, cranberries. Well, the cranberry industry started in Massachusetts in the mid-1800s when some tricks were there. They grow wild there. And they grow wild actually all the way down into the Appalachians. And it spread from Massachusetts to other parts of the country. New Jersey has a not insignificant cranberry industry. The biggest, though, is in Wisconsin. Northern Wisconsin particularly has that's where the largest concentration of cranberries in the world is, actually, in Wisconsin. Then also on the West Coast, in the on either side of the Washington-Oregon border, and then up around in Canada, up around Vancouver, and also some in Quebec and other parts of eastern Canada.
Do you hire people to plant and harvest?
I do now. You don't need to plant. Cranberries last longer than any other, probably even than olive groves or things.
So once they're there, they stay. One
of our bogs, as long as you take care of them, they will get overgrown with weeds and things like that if they're not maintained properly. But at least one of our pieces, and probably a couple, we have about 10 different pieces all in kind of separate elevations and things. At least one of them is over 100 years old.
If you want to expand, how do you make the cranberries grow somewhere else?
Well, traditionally, you would build a new cranberry bog by taking a piece of wetland, building a dam at the downstream end of it with the stream running through it, building a dam at the downstream end so you could hold the water back if you wanted to, building a dam at the upstream end so you could create a reservoir behind it and drain this part. You would then cover it with sand and plant cranberries. Cranberries are planted from cuttings pushed directly into sand. Sand is what they like is a sand-water interface. That's their natural habitat. And that's where they were growing and originally some of the main varieties that are still grown extensively in Massachusetts in particular were just collected from the wild on Cape Cod and propagated vegetatively by putting cuttings into sand. So
how do you keep water in it?
So the bog doesn't need to have water all the time.
I see.
Cranberries actually grow on dry land, as it were, although they need fairly frequent irrigation. They don't like to have really wet feet. They need very well drained sandy soil. They grow in pure sand. Actually, if you showed a corn farmer a cranberry bog about to be planted, he'd laugh his head off because it just looks like a parking lot.
You have to fertilize?
Oh yeah,
you do.
Yeah, so you have to fertilize. There are lots of pests you have to deal with. There are weeds you have to deal with. There's all that stuff. But their growth medium is basically pure sand. And then an important cultural technique is every few years you have to spread about 2 centimeters of sand on top of the plants or on the plants to suppress weeds, to encourage new growth, and so on. Sand is like fertilizer to them. Actually, if there's a bunch of cranberry plants growing, and there's a sort of a bare sandy area, they'll immediatally send runners right into it.
And how do you harvest them?
Cranberries are harvested by flooding the bogs. There are 2 processes. One is a dry process where you have a picking machine that goes through them. It's fairly laborious, although that used to be done by people on their hands and knees with scoops, which was really laborious. But it's the most cranberries are harvested with a so-called wet pick method, where you flood the bog with water, and then you run over it with a beating machine. That beating machine knocks the cranberries off, and cranberries are hollow.
They float.
They float, and so it's really quite pretty to watch the cranberries popping up in this on a nice day with the blue water and trees changing color in the background and so on, and popping up in this big streak behind the beating machine. You then surround that with a big plastic flexible plastic boom, pull them all to one side, and they now go through a rather cleverly designed pump system into a truck, and the truck then can go off to the receiving station from which they get processed into juice or sauce or whatever.
And they weigh them, and they pay you for how much weight you give
exactly. When they weigh them, they take a sample.
Nice.
They as the truck goes on the scale. As it goes in, sits on a scale, and while they're getting the weight, they put a probe in at random places and pull out a sample, and then they clean and dry that sample, and they weigh it, then clean and dry it, weigh it again, and proportion the whole weight of the load to what the yield they can determine from the sample. They also test for color and a few other things at the same time. And
I have a bag of cranberries and dates mixed.
There you go.
I love dried cranberries.
Good product.
They're great.
Good product. Ocean Spray.
So did you harvest this?
Yeah, a couple of weeks ago.
It's done. And you go out and help out, or?
Oh yeah. While I'm there, I manage the water. Is the main thing I do. We also have a big party though. Next time, I'll send you an invite next year. Have a big party. Yeah. One of the days to harvest.
Every time I've heard you give a talk, you always mention your cranberries.
Right.
You must like it a lot.
I showed the slide
today. Yeah. I have to get that picture. We'll use it as the episode art.
There you go. I'll send it.
But you enjoy it, right? It's relaxing.
Oh yeah, it's different. It's using different muscles, as it were.
Nice. All right. Well, that's great. Well, on that note, this episode of This Week in Virology you can find at TWiV.tv, also on iTunes. And if you have any questions or comments, please send them over to TWiV at TWiV.tv. My guest today has been John Coffin from Tufts University, also the National Cancer Institute. Thanks so much for talking with me today, John.
You're very welcome. It's been my pleasure.
Appreciate it. Great story. It's good to hear virology history. Not many people know it anymore.
It's true.
And it needs to be recorded forever because, there's a lot of good stuff going on today, but most of the people, and they're very good who do it, have no idea what happened. And I think it's instructive to know where even little things like using half a curie of P32 at one point, how far we've come now where we don't have to do that anymore.
Yep, that's certainly true.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.